Well, hello. Just while we wait for the technical things to catch up, I'd like to introduce myself. I'm Laura Denby. I'm a research scientist, so I'm one of these supposed boffins that lives in a lab, wears a white coat. And it's an absolute privilege for me to be here today because I don't get to see patients because I work in a laboratory, and it really is a privilege to get to speak to you today here in the room and on Facebook. So hello everybody there. So I'm going to do a mixture between showing some slides and just talking to you, because the problem with what I'm going to talk about is it's quite complicated, so hopefully I'm going to drill it down to quite a basic level so I can take you all with me and you can sort of understand the research that I'm doing. So I think my title was, what does scientific research tell you about IgAN? Well, in order for you to follow my talk, I need to introduce DNA. So hopefully you all know what DNA is. And you hopefully understand that it essentially is the recipe of you, if you put it very simply. It contains all the information that makes me, me, you, and that is what we need to know about it. And we know hopefully that within that you have genes. And these genes encode specific information or a message that will make every single building block that makes your cell and your whole body. Okay? Now, we're going to play a quick game because I always think talks need to be exciting. So I'm going to quickly grab this banana here and ask you, how many genes do you think this banana contains? Here's the banana, quite simple. Come on, have a guess. Anybody? 10 genes. Is that what you're going for? Yes. Any advance on 10? We're going for 10?
I'd go for a couple of thousand.
Couple of thousand? Okay, what if I tell you that it's about 35,500 genes? 35,500 genes in this banana that makes this banana like this. Okay? Another one. This packet of rice. How many genes do we think rice has? Bear in mind the banana has 35,500 genes. Any suggestions? Anybody? Higher or lower even? Higher, lower. Oh, a mixture. Essentially, it's a bit higher, up to 52,000 genes. Okay? So that's banana and rice. So, humans, how many genes do we have? Million. We have just over 22,000 genes. We're nowhere near as complex as rice and bananas, thinking we're top of the food chain there when we're not. But this is quite important. So, we're not got as many genes. But actually, we're far more sophisticated than a banana and rice on a genetic level in our DNA. And this just over 22,000 genes, actually only covers 1.2% of your DNA. So this information is only on 1.2%. What on earth is the other 98% doing? Well, as scientists, we decided to call that junk DNA. Wasn't doing anything, it's just there, junk, ignore it. We're only interested in these things called genes that make messages, that make the building blocks of our cells and our bodies. Since the DNA revolution, where we've taken part and we're able to actually sequence people's genomes now, what we've actually discovered is that about 70% of this DNA actually has a signal coming off it. But that signal isn't making a protein or a building block for your cells. It's doing something else. And this takes us into the realm of what's called non-coding RNA, okay? Now, typically, DNA makes a message, and we call that messenger RNA. So this is the message that's making whatever it needs to make. Non-coding RNA, quite simply, doesn't make anything. So that's quite easy to remember. But what we discovered was that this non-coding RNA is actually very important. Go back a bit. So, 11 years ago, when I was a bit younger, a little bit less white hair, I was really interested in a rat model of glomerulonephritis. So hopefully you understand that's an inflamed glomerulus, but in a rat model, because I'm a scientist, I'm not a doctor or a clinical doctor. And I became really interested in trying to understand if this non-coding RNA, that really was quite new and exciting, was important in a model which basically mimics IgA nephropathy. And what I found was that actually it was. And the particular types of non-coding RNA I'm interested in are teeny tiny pieces of non-coding RNA, just they're very small, so they get called small non-coding RNA. We're very good at describing things in science. Basically what they are is they're interceptors in the basic form. So these teeny bits of RNA, they intercept the message that makes the building blocks and stop the building block being made. Okay? So they're blocking the message. And in normal, healthy tissue, they're very important because what they do is the cell goes, oh, I maybe need a bit more of this sort of building block. So they'll change the level subtly and a bit more will be made or a bit less will be made. However, what I found was that in this rat model, and in fact a whole host of renal models of disease, that these little interceptors are very abnormally expressed. So essentially these little molecules are very high, so they're having a huge effect on the ability of your cell to make what it needs. And the one that was the most interesting was a microRNA called miR-214. That's its official title. And this became essentially the focus of my work for about a decade. And it's really interesting because it's upregulated in your kidneys in every preclinical model that I've looked at. So it started off in an IgAN type model, but actually it's true in other renal diseases as well. And I went the whole way through showing it was different, and I actually got a drug company who made a drug that could block this teeny tiny little microRNA. And this picture here is a bit of real science. So this isn't a human kidney, this is a mouse kidney, okay? So what I've got here is your normal mouse kidney, and you can see hopefully it's yellow. Okay, can you see okay over there? When we injure the kidney, which is what I've done here, you can see it goes red. Now that red staining represents scarring. So I don't know if you know this, but as your kidneys start to get more and more damaged, they get more and more scar tissue inside them, and this scar tissue is bad news because it replaces your normal, healthy, functioning nephrons that are basically filtering your pee normally. And if you lose more of these, your function obviously declines. So red, even in mouse and in human kidneys, is bad news, and we want to try and stop it. And here in this last panel, hopefully, you see, it's basically yellow, looks very similar to this. And what this little drug was able to do, when given to this mouse, was drop the amount of scarring by about 86%. And that's really exciting. It's a huge reduction. And I've continued to sort of work on this. But the next question was, well, this is in mouse, so what? We need to show that it's important in patients too. So here I've just got some kidney biopsies that John has shown you previously. At the top, we've just stained them with a stain that lets us look at what they look like basically. So hopefully you can see here, we've got a glomerulus, the wee sieves, and we've got the little tubules. In this panel here, I've got purple staining for the microRNA that I'm interested in. And I'm going to make this bigger so everyone could see it. Hopefully you can see that here is very strong staining for this particular microRNA. Here are your tubules that are actually injured ones. You see they're sort of flattened. They don't look like these ones. They've got a really high signal. And also importantly, in the inflammatory cells, which is what this lower panel here is, we have a very high signal coming from there. So 214 is upregulated in the biopsies of patients who have IgAN and other renal diseases. As I said, I'm a basic scientist, so I was really happy when another group went forward and had a look at actual human biopsy samples. So we've already talked about biopsies today, but this group got some RNA from these biopsies and had a look for this particular little molecule. And what they found was that if you had a large amount of scarring, then you had higher levels of this particular little microRNA 214. And even more so, the expression of this 214 little molecule, the higher it was, the higher risk of mortality due to renal failure. So this little microRNA, if it's present in your kidneys, is not good. And this is a new thing. So we've only known about these little molecules for less than 20 years. So this is a step forward of new things and new ways of being able to look at kidney disease that we previously didn't have. So rather excitingly, I'm still trying to get this translated and looking for ways to sort of move this into more clinical sort of testing. But one of the other things we found when we were doing this work was that these little microRNAs, these little bits of RNA, are actually in your blood and in your urine. They're really stable, and they're really easy for us to extract and to measure. So we thought, well, at the moment, patients have to have a biopsy in order to be able to tell what's wrong with their kidney. Is there any way we could consider moving forward? Is there a blood test or a urine test we could do that maybe would reduce the number of people who needed biopsies, or even just be able to give doctors a little bit more information about your disease? For example, is it progressing really fast? You may feel okay, but actually your kidney is actually not doing so well. So we started on this project, and I would like to thank any of the patients here who are from Edinburgh and Glasgow that have participated in this study. But we collected patients primarily with IgAN, blood and urine, to ask the question of whether or not these little tiny bits of RNA could actually tell us about their condition. And the reason we sort of started that was, if I take this little graph here, it's a bit boring, I know a lot of my work is just graphs, but here along this one we have the levels of 214 that you've got expressed, and here's the level of fibrosis. And even more confusingly, lower is higher in this particular graph. So the patients who had high levels of miR-214 had much higher levels of scarring within their kidney. And now this is quite important, because that could tell your doctor that maybe, you maybe need to be seen a bit faster, or they need to look at you a little bit closer. So we set out this sensor study, which was called Non-Invasive Biomarkers of Renal Disease. It was done between Edinburgh and the Royal Infirmary and in the Queen Elizabeth University Hospital here in Glasgow. We've recruited over 750 patients, primarily those with IgAN, and we've collected 200 patients who came in and have just had their biopsy in the last couple of years. And at the moment, we're busy analysing all this data to see whether or not these particular microRNAs can, one, tell you that you have IgAN as opposed to any other condition, and also whether or not there's a chance that the levels of these could maybe tell you about whether or not you'll have a maybe more rapid progression in your disease, or maybe if your disease will be sort of more slowly, and it won't really do much over a long period of time, because this is important information for your doctors to know to make sure you're getting the best sorts of care. And I thought I would treat you to some new data that was actually done in collaboration with John, who's just spoken, because we're a community, we work together for your benefit. We don't work in isolation. We're all working hard to try and find new things to help you in your disease. And Leicester has one of the largest collection of IgA samples that's very well phenotyped. And here we've got some new data from this sort of microRNAs that we're measuring. So this graph here is actually just the housekeeper, because even in science we need housekeepers, and that's to make sure that our data is neat and tidy and is comparable with all other data. So here's just looking at one particular microRNA, 400 patients, and hopefully you can see its expression is very static and is basically a straight line, which is what we'd like to see, because we don't want that one to move. Here on the left-hand side, we've actually got one of the specific microRNAs that I've found to be different between normal people and people who have IgAN. And hopefully here, you can see it's just 24 patients as an example, that we have a far more variable expression in these microRNAs in the blood. So therefore, what we're doing is we're doing sort of fancy mathematical statistical analysis now to see whether or not, for example, the patients that have higher expression, what does that tell us about them? What do we know? Maybe tells us a little bit more about their condition compared to the other patients. And this is very exciting, going on here in Scotland, and hopefully we'll have the results of this finished towards the end of the new year. But what's really exciting on the basic science side of things, which I really wanted to tell you about, was we're at basically the era of the omic-led research. That doesn't really mean anything omic, but essentially it's telling you that we've now created such fancy technology that we can now look at whole kidneys and see how they're different in disease and normal healthy tissue. We can go a step further, take the kidney and take out just the individual cells that make up the kidney and ask, how are they different in disease and in normal tissue? And even more recently, go another step to single cell technology. So we can separate your kidney into individual cells and look at the expression of this messenger RNA and these genes in an individual kidney cell. And this has been done in mouse and in human and is really so exciting. But I can tell you how fast this is progressing. 5 years ago, this was cutting edge, whole kidney sort of examinations. In 5 years, we have gone down to a single cell level. Now this is obviously in mouse, this tissue here, this is your lovely kidney of a mouse, all separated, but we are able to do it now in biopsy tissue from patients. And what this will allow us to do is understand your disease on a level that we've previously not been able to do and learn far more about each individual disease, which should allow for a quicker generation of new therapies, but also perhaps in the future lead to more personalised medicine so that you're getting the best medicine for your disease. And I think this is all really, really exciting. And I want to show you that it's happening in Scotland. We're doing all this sort of thing. We're collaborating with the world leaders such as Sean, who's able to translate these sorts of findings to the clinic and into patients. So I think the future is really bright in terms of a revolution in how we're able to diagnose and treat patients in the future. I also just want to throw this in because Scotland is here. We're a big country here. There we go. But we actually have, and we're very fortunate to have 4 independent basic science fellows. So these are research scientists. We have 4, 2, 1 in Glasgow and 3 in Edinburgh currently, which is actually, of the whole basic science fellows, is actually a very large amount. And also, very importantly for me, we're all women. So Scotland's bucking the trend. We're getting women into science, we're getting them up, we're getting them fellowships. And I think that's really important, that we've got role models for the next generation of children coming through to say, do you know what, I can do this, I can become a scientist, which is really exciting. Finally, acknowledgements. I have a huge list here to thank. Lots and lots of nephrologists here in Scotland that you may or may not know, as well as all my basic science people. This is my lab, a small team made up of basic scientists and also trainee transplant surgeons, because we all work together, as I said. And this is a sunny day in Edinburgh, because honestly, in Scotland, we all know it's sunny every day. Thank you very much for your attention.